Hi, everybody. Dr. Becker. We're moving on now to Chapter 4. This is a two-part series for this lecture. So Chapter 4 is the cell physiology and pathophysiology. And again, this is Part 1. There's a lot of content as well in this section. It goes right into key concepts, again, that I would read through at the end. There's 4 slides just of the key concepts. So we'll come back to that at the end. Essentially, we're going to talk about the cells in the body perform many different functions. And the focus of the chapter then is the physiology and pathophysiology of how the cell functions, how the cells cross the membranes, how the substances affect the cell signaling, etc. So the first part here is diving into the cellular components. It's a good place to start. So before we go any further into the text and what the text talks about, let's go ahead and watch our osmosis video. Remember, the osmosis content is not in and of itself on your exams or quizzes. This is a review. There's going to be some topics on here that maybe we don't actually address in your text, but it's a good review, and you can come back and listen to this again at any time as well.
The cell is the basic unit that makes up every living organism. It's the smallest form of life that can replicate on its own, but cells in our body differ quite a bit from each other. The human body alone has over 200 distinct cell types, from long skinny neurons that can go over 1 meter long, to macrophages that gobble up pathogens, to myocytes that contract to let you flex your muscles. But despite their differences, they do share a lot of similar features. So let's imagine the average cell as a small apartment. First, we want some walls to distinguish between what's outside and what's inside. These walls are the cell membrane, or plasma membrane, and they're made out of a double layer of phospholipid molecules. Phospholipids have a head made out of negatively charged phosphate, which is hydrophilic, meaning it likes water. Phospholipids also have a tail made out of 2 fatty acids, which are hydrophobic, meaning they avoid water. In water, phospholipids form a bilayer, where the hydrophobic tails are oriented inwards, where there's no water, and the hydrophilic heads orient themselves outwards, in contact with water molecules. So the plasma membrane forms a wall with water on both sides. This wall is semi-permeable, meaning it allows some things to go through, like oxygen or carbon dioxide, but does not allow other things through, like glucose and sugars. Fortunately, we have the doors and windows on this apartment, and they're made of special protein channels that are essentially tiny tunnels through the phospholipid bilayer. These channels allow water and specific ions like sodium and potassium to come in and out of the cell. Now, just like any well-built apartment, our cell has a sturdy framework called the cytoskeleton. The cytoskeleton is made out of proteins like microfilaments, microtubules, and intermediate filaments, which all provide structural stability. The cytoskeleton is also very dynamic, allowing the cell to change shape by selectively contracting and extending filaments, which is important in some cell functions, like muscle contraction, cell division, and even cell movement. The cytoskeleton also helps structures within the cell move from one area to another. Now that the apartment is built, let's fill it in. The cell is filled with intracellular fluid called cytosol, which contains various ions like sodium and potassium. In addition, cells have a number of organelles, which are like little rooms within our apartment. And together, the cytosol and the organelles make up the cytoplasm of the cell. Now, the nucleus is a highly specialized organelle, and is home to our genetic material. In terms of rooms, you can picture the nucleus as the office, with bookshelves containing various books and manuals. How to do home repair, how to do interior remodeling, how to talk to your neighbors, how to take out the trash, and so on. Extending the analogy, each book would be like a gene that contains the exact information, kind of like a blueprint, that's needed to build a specific protein. Drilling down deeper, the exact letters and words of the books are written in the language of DNA. Now, as it turns out, DNA is actually one long molecule with some parts that code for genes connected together by other parts that don't code for genes. If the DNA molecule in a cell were stretched out, it would be about 2 meters long. Fortunately, the DNA is slightly coiled and wound in multiple steps so that it packs down really tight. First off, it's coiled around proteins called histones. Zoomed into that level, the DNA looks like pearls on a string. But then that necklace gets folded further and further, creating an extremely neatly folded and tightly packed form of DNA, called a chromosome. Many chromosomes create chromatin, which is a shapeless mass of DNA floating in the nucleus. Now, to make a protein, the DNA of a particular gene has to be transcribed or copied over into RNA, specifically messenger RNA or mRNA. Special structures inside the cell called ribosomes can then use the mRNA copies to produce proteins. The ribosomes are made by a structure within the nucleus called the nucleolus, out of ribosomal RNA or rRNA and proteins. They're like the workshop of our cell apartment, with mini 3D printers, printing up various spare parts or tools for the apartment, which is the analogy for protein synthesis. The ribosome converts mRNA into a string of amino acids, which forms the protein in a process called translation. Ribosomes rely on a triplet code, where every 3 nucleic acids along a sequence of mRNA correspond to one of the 20 common amino acids, or to a stop codon, which tells the ribosome to stop building the protein. So to help visualize all of this, imagine the ribosome floating around in the cytosol. Once that ribosome binds to mRNA, it uses the cytoskeleton to help make its way over to an organelle called the endoplasmic reticulum, or ER for short. The ribosome attaches to the plasma membrane surface of the endoplasmic reticulum, and will start translating the mRNA into a protein that gets directly injected into the lumen, or inside of the endoplasmic reticulum. The endoplasmic reticulum supports lots of ribosomes on its surface, each of which is busy translating proteins. To create enough surface area to do this, the endoplasmic reticulum is really a continuous stack of flattened phospholipid membrane. Sort of like a huge deflated balloon that's all crumpled up. It's low in volume, but it has a lot of surface area available. The part of the endoplasmic reticulum that produces protein is called the rough endoplasmic reticulum, since the ribosomes resemble tiny little bumps that make the endoplasmic reticulum look rough under an electron microscope. There's also a smooth type of endoplasmic reticulum, which is involved in making lipids, like cholesterol and phospholipids. And the cell can use this for producing more cell membrane. In some cells, like the liver, the smooth endoplasmic reticulum helps with detoxifying harmful chemicals, so in this case, kind of like a spa or bathroom in the apartment. In other cells, like glands, the smooth endoplasmic reticulum uses cholesterol to go on to create various steroid hormones. For this reason, some specialized hormone-producing cells, like the ones found in the testes or the ovaries, have lots of smooth endoplasmic reticulum. Another organelle called the Golgi apparatus takes the proteins, lipids, and hormones that are generated in the rough and smooth endoplasmic reticulum and packages them into a vesicle made of a double layer of phospholipids that are then distributed around the cell. Think of the Golgi apparatus like the garage of the apartment, where you have all your packed up boxes that you sometimes then move and store in other rooms. There are transport vesicles which move within the cell, and secretory vesicles which move molecules out of the cell. A process known as exocytosis, kind of like taking the garbage out of the garage. An example of a vesicle that stays in the cell would be the lysosome, which contains enzymes that can help break down other molecules, as well as large organelles that might be damaged. Finally, there are the mitochondria, which are organelles that serve as the power generators, like in the utility closet of our apartment. In the cytoplasm, glucose goes through glycolysis, which is a metabolic pathway that cleaves the 6-carbon glucose into 2 halves, which are 3 carbon molecules called pyruvate. From there, the pyruvate enters the mitochondria and goes through the citric acid cycle or Krebs cycle, as well as the electron transport chain, in order to produce adenosine triphosphate, or ATP. These processes require oxygen, so this is termed aerobic respiration. The byproducts of this set of reaction are carbon dioxide and water. Ultimately, about 32 molecules of energy-rich ATP are made for every single molecule of glucose taken in by the cell. Alternatively, if there's not enough glucose to go around, our cell can try burning fatty acids in the mitochondria as a source of fuel, in a process called beta oxidation. Now, it turns out that mitochondria can only work with medium-sized fatty acids. So to make it work, a special organelle called the peroxisome chops long fatty acids down into medium-sized ones. The process generates dangerous hydrogen peroxide, but the peroxisome has an enzyme called peroxidase, which safely converts the hydrogen peroxide into water and oxygen. Alright, as a quick recap. The phospholipid bilayer forms the cell membrane, which are the tiny walls of the cell, and the internal structure of the cell is made up of a cytoskeleton and cytoplasm. The key organelles include the nucleus, which houses the DNA, the endoplasmic reticulum, which generates proteins, lipids, and steroids, and the mitochondria, which provides the cell with energy using aerobic respiration, which burns glucose, or beta oxidation, which burns fatty acids.
Osmosis video is very helpful to kind of outline and have these components make sense. I like that they break it down into that apartment example. So in your book, these cell components are outlined. I do think it's worth it for you to write out the definitions and understand, what they are. As far as the cell membrane goes, it's really important when you think about it because that is the layer or the boundary between your intracellular and your extracellular fluids. We can call this the plasma membrane. It's a phospholipid bilayer, and that video did a great job of showing that it orients itself specifically to have the hydrophilic towards the outside that likes water, and it keeps the hydrophobic components towards the inside of the cell, which is very important to understand. The non-polar fatty acid tails of both leaflets will make up the hydrophobic core of the membrane, and that's that critical barrier that's going to separate the intracellular from extracellular fluid. The molecules of cholesterol are interspersed within those phospholipids, which helps control the membrane fluidity. So in this book, it talks about if you picture the cell membrane as a grilled cheese sandwich, with the outermost bread layers able to mix with water and the polar solutes, but the melted inner core is acting like a barrier impenetrable to water and polar and charged solutes. We talk a lot about things crossing the cellular membrane or the plasma membrane. So integral proteins are needed to help with this process. Some function as receptors and others as transporters or channels. The peripheral proteins often attach to the inner leaflet and work with integral proteins to alter cell function. So your outer leaflet is rich in glycoproteins and glycolipids, which we learned in the last chapter, literally is what it sounds like, part sugar, part protein, and part sugar, part lipid. And then with the complex branching carbohydrates attached. Okay. The book specifies that the layers of sugars projecting outward from the cell is referred to as its glycocalyx, which functions in cell-to-cell regulation. Okay, so it's really important to understand how that cell membrane works and how it's going to act as a barrier between the intracellular and extracellular fluid. Then it talks about your cytoplasm, which is the entire contents of the cell, which is made up of the cystosol, which is the other word that we learned for intracellular fluids, the organelles, and then the other structures. The aqueous intracellular fluid contains dissolved electrolytes. Remember, when they get into water, the bonds would dissolve, and they go into individual electrolytes and their small biomolecules. The cytoplasm also contains the movement protein myosin and actin in muscle cells. So within the cytoplasm, a lot of things occur. The protein synthesis occurs in the cytoplasm. We've talked a lot about the nucleus, and the book talks about this. It's holding most all of our cells' DNA, and some of the DNA is found in the mitochondria. The nucleus also contains a nucleolus that functions in synthesis of the ribosomes. So unless a cell is preparing to divide by mitosis, the nucleus contains DNA organized in chromosomes that are wound around those histone proteins. The nuclear membrane has two layers with relatively low permeability. So the nuclear pores, though, allow the movement of proteins, signaling molecules, and the messenger RNA molecules between the nucleus and cytoplasm. So red blood cells do not contain nuclei. Skeletal muscles have multiple nuclei. Other than those two things, each of the body cells contain one nucleus. Box 4.1, I think, is a good thing to review for the cell membrane features. Okay. Remember, the cell membrane can also be called the plasma membrane, and in future courses, you can hear it referred to both ways. But the cell membrane is a complex phospholipid and cholesterol bilayer. Remember, the hydrophilic or polar, the water-happy regions of the phospholipids and the cholesterol face outward to the extracellular and intracellular fluids. And the central fatty acid and cholesterol ring core that is hydrophobic resists the passage of other polar or hydrophilic solutes. So the grilled cheese sandwich in here helps that this can interact with the water and this can interact with water solutes, but it's not going to allow things to easily diffuse across the membrane, and they have to be done essentially actively with purpose, right? So we need to be guided or have channels or whatever the case may be. The membrane is studded with proteins, which is an integral part because those proteins span the membrane and they're attached to the inner and outer membrane. That's what can help things move across. Think about it that the membrane is dynamic. It's moving with motile cells, such as white blood cells. They're conducting exocytosis and endocytosis, and they're enlarging with cell hypertrophy. The clustered lipids shown in red in this figure here represent a lipid raft, which is a region of tighter lipid and cholesterol packing that tends to move as a cohesive unit. A protein is linked to the lipid raft by a GP1 anchor, allowing the protein to be associated with the plasma membrane without actual insertion through the membrane. I always referred to that as the GP1. But now that I look at it, I wonder if it's trying to say GPL. But I'm pretty certain it was GP1. All this time, I wonder if I've been saying that the wrong way. Here, it does talk about the sugary extracellular coat due to the glycoproteins and glycolipids, and that we still have those specialized proteins that'll attach to the lipid rafts. Continuing on with other cell features and functions. So organelles, the plasma membrane boundary separates the intracellular fluid, the cytosol, from extracellular fluid. So the membrane-bounded organelles include the nucleus and the mitochondria, rough and smooth endoplasmic reticulum, the Golgi apparatus, and the vesicle budding from the Golgi, lysosomes, peroxisomes, and vacuoles. So this is way taking us back to the time of chemistry. The ribosomes and the rough endoplasmic reticulum are outlined in your book, and I would understand, write out what those definitions are so you can remember that. The endoplasmic reticulum is contiguous with the nucleus, and it's the site of the synthesis of proteins that will ultimately be secreted from the cell or moved to a specific cellular location. So the endoplasmic reticulum is also very important. Within the rough endoplasmic reticulum, the proteins fold to assume, again, their secondary, tertiary, and if applicable, their quaternary structures like we've talked about in previous chapters. So when we talk about what shape the proteins fold into, this often happens within the rough endoplasmic reticulum. It's also the site of chaperone proteins that detect the progress of protein folding and that mark misfolded proteins for destruction through the unfolded protein response. The smooth endoplasmic reticulum is an intracellular membrane, and it connects to the rough endoplasmic reticulum, and it contains enzymes for all of our metabolic processes. The smooth endoplasmic reticulum is particularly abundant in liver cells, and that's really important because the liver cells do a great deal of our metabolic processing of lipid molecules, and the liver cells also conduct the biotransformation of drugs and toxins. So having the smooth endoplasmic reticulum and where it's located is very important. Some of the sites it's located in also are involved in some of the steps in steroid hormone synthesis. It's also the site of synthesis of new membranes that can refresh the cell membrane or organelle membranes. Okay. Smooth endoplasmic reticulum is a major site of cellular calcium homeostasis. In muscles, the smooth endoplasmic reticulum is abbreviated SER, but it's also called sarcoplasmic reticulum in the muscles, and that functions to release the calcium ions that initiate muscle cell contraction. So it subsequently takes up the calcium ions and causes muscle cell relaxation. The Golgi apparatus is a stack of membranes organized in the flattened sacs. It functions to sort, modify, and package proteins and lipids that are then delivered in the vesicles from the nearby endoplasmic reticulum. The mitochondria has the two layers that produce ATP through the Krebs cycle. That should be ringing a bell. And oxidative phosphorylation. This brings us back to chemistry, right? So the mitochondria are always referred to typically as the powerhouse. That's how I also learned it in undergraduate, because they produce much of the cell's adenosine triphosphate, the ATP. And if we remember, ATP is a great prime energy source for cellular chemical reactions and ion transport. And they're the source of phosphate for the phosphorylation reactions. So energy-producing reactions of the Krebs cycle and oxidative phosphorylation take place in the mitochondria. So they are the sites of cellular respiration. They consume the oxygen and then generate the carbon dioxide. Cells with the highest energy requirements have a correspondingly higher number of mitochondria. So the cells that have the highest output need the highest energy. So they're going to have the most powerhouses. Other significant aspects of your mitochondrial function are listed in your book, and that's when it talks about they have those two membranes, and some of the mitochondrial functions occur within the matrix, while others are in the inner membrane. Mitochondria also contain their own DNA that codes for several of the mitochondrial proteins. Some steps of the steroid hormone synthesis and of fatty acid catabolism take place in the mitochondria. And the mitochondria are able to sequester calcium, so they play a role in initiating apoptosis, which is programmed cellular death. So the mitochondria are busy and they're very important. Lysosomes contain degradative enzymes, the proteases and the phospholipases. So they are really important in breaking down aging cell organelles and molecules. The degradative enzymes are called acid hydrolases, and they depend on low pH for their function. Some examples of the acid hydrolases include proteases and phospholipases. The proteases degrade proteins, and the phospholipases degrade phospholipids in their membrane. Some of our phagocytic cells, such as our neutrophils and phagocytosis of bacteria, is followed by fusion of lysosomes, forming a phagolysome. The lysosomal degradative enzymes then contribute to killing bacteria. We talk about this in your next course in advanced pharmacology. How do antibiotics work? So the lysosomes are very important in a lot of bacteria, and antibiotics work through the use and manipulation of lysosomes to seal off bacteria. The lysosomes also contribute to cell death pathways. The peroxisomes, they conduct your oxidative metabolic reactions. They can generate and detoxify reactive oxygen species, such as your hydrogen peroxide. Your cytoskeleton is made up of your protein filaments that extend throughout the cytoplasm, many of which are anchored to your membrane proteins, and this is what gives the cells its shape. So the filaments can be organized by size from small to large, like microfilaments made of actin, intermediate filaments composed of several different proteins, and then the microtubules made of tubulin. So organelles and vesicles can move along these filaments, and in motile cells, especially like white blood cells, they are responsible for the cell movement. Principles of membrane transport is kind of the next topic your book goes into. So before we jump into that, let's watch our osmosis video. It kind of repeats some of the things we've learned already, but also helps us understand membrane transport.
Side of a cell.
Let's start that.
The intracellular space.
This
Versus what's on the outside of a cell, the extracellular space. And it helps regulate what comes in or out of the cell, which is called selective permeability. Movement across the cell membrane can happen by passive transport, which requires no energy, as well as active transport, which requires energy in the form of adenosine triphosphate, or ATP for short. There are 3 types of passive transport: diffusion, facilitated diffusion, and osmosis. Diffusion helps small, nonpolar molecules like oxygen and carbon dioxide move across the membrane, from an area of high concentration to low concentration. The difference in concentrations is known as the concentration gradient, and diffusion can happen as long as this gradient exists, and stops once there are equal concentrations of the molecule on both sides. Facilitated diffusion helps larger molecules and polar molecules move across the membrane. Facilitated diffusion uses transport proteins like channels and carrier proteins. Channels are not very specific, and they can open or close to allow water and small polar molecules, like ions, that are dissolved in it to pass through. These channels open in response to certain stimuli. For example, the voltage-gated calcium channels respond when the electrical charges on the two sides of the cell membrane change. Normally, there are more negative ions inside the cell than outside. So the inside of the cell membrane is negatively charged, and the outside is positively charged. When positive ions make their way inside the cell through one form of transport or another, the cell starts to depolarize gradually, so step by step. The membrane becomes more positive on the inside and more negative on the outside. This change in the distribution of electrical charges makes the voltage-gated calcium channels open and allow water, a lot of calcium ions, and a few sodium ions inside the cell. When the entire membrane is depolarized, the channels close back up and inflow stops. In contrast, carrier proteins are very specific. They have special binding sites that only allow certain molecules to bind to them, and they also have gates at both ends, which open sequentially. An example of a carrier protein is the glucose transporter protein called GLUT4. By default, the transporter is open to the outside, waiting for a glucose molecule to float by. When a glucose molecule binds to a special site within the transporter, the outer gate closes, and the inner gate opens. The glucose then unbinds and floats freely into the cell, while the transporter will reset, awaiting for the next glucose molecule. Since glucose is moving down its concentration gradient, this requires no energy. Finally, there's osmosis, which, in addition to being an awesome company that explains medical topics really clearly, also describes the movement of water across the cell membrane. It's a special case of facilitated diffusion, because water uses channels called aquaporins to move across the membrane. Osmosis is driven by osmotic pressure, which is a force determined by the difference in solutes between the two sides. Osmotic pressure drives water from the hypotonic side, or the one with lower concentration of solutes, to the hypertonic side, or the one with higher concentration of solutes. But sometimes molecules need to be transported against their concentration gradient. That requires energy, or active transport across the cell membrane. Now there are two kinds of active transport, primary and secondary. Primary active transport directly uses ATP as fuel, and it's done by a family of enzymes called ATPases. ATPases have the ability to pump out substances, like ions, using ATP. Sort of like a candy dispenser. You insert a coin and candy pops out, or in our case, an ion pops out. The most important example is the sodium-potassium ATPase, which is a double-action pump, meaning that for each ATP molecule, it pumps 3 ions of sodium out of the cell, while at the same time pumping 2 ions of potassium into the cell. This is an example of an antiport, because the two species of ions are pumped in opposite directions across the membrane. The resulting difference in concentrations of ions on either side of the membrane creates a concentration gradient. But since ions also carry electrical charges, it's considered an electrochemical gradient, and the cell can use this in secondary active transport. Secondary active transport does not directly require ATP, but uses an electrochemical gradient, just like the one made using the sodium-potassium ATPase. An example is the sodium-glucose SGLT1 transporter, which pulls a molecule of glucose into the cell against its concentration gradient, along with 2 sodium ions that move along their concentration gradient. Since both sodium and glucose go in the same direction, this transporter is called a symport. Now, in addition to pulling in individual molecules and ions, sometimes cells need to take in relatively large substances, like proteins, fat droplets, and sometimes entire other cells. In these situations, the cell resorts to bulk transport, using endocytosis, where the cell membrane invaginates and pulls in something from the outside. Or using exocytosis, where a vesicle from within the cell pushes something out. Alright, as a quick recap, passive transport requires no energy, and can happen as diffusion or facilitated diffusion, which relies on carrier proteins or protein channels. There's also osmosis, which is when water moves from a hypotonic concentration to a hypertonic concentration. Osmosis is considered a special case of facilitated diffusion. Active transport, on the other hand, requires energy, and can either happen as primary active transport, which uses ATP, or secondary active transport, which relies on concentration gradients.
That's another fantastic review from Osmosis. It really helps explain this, and I think in a very easy and simple way that's nice to see illustrated like that. And those principles of the membrane transport are essentially what is listed here. Oh, I did want to point out too that one of the big things to remember about membrane transport as well is that the extracellular and the intracellular concentrations of some of the solutes are listed in your table 4.1. So they point out the striking difference in sodium and potassium. Sodium is high extracellularly and potassium is high in the intracellular compartment. But the concentration gradients then for sodium and potassium what drive many of our transport processes. And so it's really important to understand how the sodium and potassium transports play so much of a role in membrane transport. The concept of diffusion then is whenever a substance will diffuse from a high area of concentration to a low area if there's no barrier. I always think of when you drop a drop of food coloring in a glass of water, it starts off very dark red, and then as it diffuses across the length of the glass, it turns to a light pink. This is the same concept for diffusion. So remember that it's a down gradient. It's going to go from high concentration down to low concentration. And this happens within our cells as well. So to diffuse through the cell membrane's lipid core requires a substance that is relatively small with a hydrophobic non-polar chemical structure. Net diffusion from one side to another is, again, a downhill direction, and it's the response to the concentration gradient. They give the example of oxygen and carbon dioxide, with oxygen entering the tissues from the capillaries, diffusing into cells, and being continually used for the cell metabolic processes, so lowering its intracellular concentration as it's used up. So it's favoring a continual diffusion into the cell, because within outside of the cell, the concentration of oxygen is higher, and then it naturally gravitates to lower areas, and the area within the cell is going to constantly being used up, so it stays low, and it naturally is going to cross that gradient. So it talks about that here in this concentration gradient example. So in this one, it is showing that the lipid bilayer has the plasma membrane. Our extracellular fluids are high concentration of these oxygen molecules. So it goes from high down to low, right? And then as these get gobbled up and it comes higher on and these get used up, then that cycle continues. This one here is concentration gradient across a barrier, permitting movement from high to low concentration again. So a selective permeable membrane, high concentration over to low, kind of this way. But then we also can have this one here, which is the concentration gradient across a barrier in which a solute needs energy to be moved from low to high. So it's an uphill movement and requires energy to get over there. The example your book also talks about is that steroid hormones are relatively hydrophobic, so they don't like water, and they will diffuse across cell membranes, binding to the intracellular receptors to exert their biological effects. Fatty acids enter cells and then can be used as an energy source through oxidation in the mitochondria and peroxisomes. Many drugs that we have are hydrophobic, and they will diffuse into cells to produce their biological effect. So they're designed that way on purpose. When substances are small, uncharged, nonpolar, they don't like water, and substances that are larger, uncharged, that don't like water, they are able to cross the cell membrane by diffusing through the lipid bilayer. All other substances, though, require a means of transport, whether it's going in or out of the cell. All right, so let's watch another osmosis video here that helps us talk about endocytosis and exocytosis. I think they're going to do a better job for you than I am of explaining this, and I like their illustration.
Cells transport material in and out across their cell membrane, which is a barrier made up of a double layer of lipids with embedded protein and carbohydrate components. Some molecules can diffuse across the membrane, or be transported across with the help of membrane-bound proteins. For transport of larger cargo, however, cells use endocytosis and exocytosis to transport material in and out of the cell, respectively. There are roughly 5 categories of molecule that try to get across the cell membrane. Small nonpolar molecules like oxygen or carbon dioxide are able to diffuse rapidly through the cell membrane. Small polar molecules like water can cross as well, but do so very slowly. Large nonpolar molecules like vitamin A are also very slow to cross the cell membrane. And large polar molecules like glucose and highly polar charged ions like sodium, potassium, and chloride, as well as molecules that possess a charge like amino acids, are all highly unlikely to get across a cell membrane on their own. So, many of these molecules, some common ones being water, glucose, and ions, pass through the membrane using transport proteins. Examples of transport proteins include channels like aquaporins, which is a water channel, and chloride channels, which let chloride ions cross membranes, as well as carriers such as the glucose transporter. However, when the cell needs to transport a lot of molecules or one very big molecule, it resorts to bulk transport, which comes in 2 styles, endocytosis and exocytosis. Endocytosis is a process that cells use to engulf extracellular material, and exocytosis is the opposite of that, during which cells expel material into the extracellular space. Both endocytosis and exocytosis need energy in the form of adenosine triphosphate, or ATP, used in the movement of the substances in and out of the cell. There are 3 types of endocytosis: phagocytosis, pinocytosis, and receptor-mediated endocytosis. Phagocytosis, where phago means to eat, is used by white blood cells like macrophages and neutrophils, which patrol the body looking for debris, bacteria, and dead cells to eat. So let's imagine that a macrophage comes across a particularly bothersome streptococcus. First, the strep attaches to macrophage receptors on its cell surface. The macrophage then extends arm-like projections called pseudopods around the strep, like a death hug. Then the strep is slowly engulfed by the cell membrane, which invaginates to form a vesicle on its inner side. The vesicle then separates from the cell membrane, forming a phagosome. During this step, an electron pump uses ATP to pump protons into the phagosome, lowering the pH inside. In the cytoplasm, the phagosome encounters an organelle called a lysosome, which contains digestive enzymes. The lysosome and the phagosome fuse together, merging their contents, forming a structure known as the phagolysosome. Inside the phagolysosome, lysosomal enzymes start destroying the bacteria with the help of an acidic pH. After it's all over, lysosome heads over to the cell membrane to expel the leftovers out into the extracellular space, like a cellular burp. Pinocytosis, on the other hand, literally means the cell drinks. In pinocytosis, the cell's plasma membrane invaginates to form a small cup around the portions of extracellular fluid and solutes that are not dissolved in it. Then, the edges of the cup come together, forming a vesicle. Since the cell is not really eating anything other than the occasional solute, the result is not a phagosome, but merely a vesicle. The pinocytosis vesicle is much smaller than a phagosome. Also, unlike phagocytosis, pinocytosis is a non-specific way for cells to take in solutes. So, whatever solutes are hanging around in the extracellular fluid get pulled inside the cup as well. And once inside the cell, motor proteins like kinesin or dynein carry the pinocytosis vesicle using ATP deeper into the cytosol. At the same time, the vesicle slowly releases the extracellular fluid and the solutes into the cytosol as well. Now, finally, sometimes endocytosis involves special receptor proteins on the cell membrane, so it's called receptor-mediated endocytosis. Some molecules are taken in this way, like transferrin, which is an iron-binding protein, or low-density lipoproteins, or LDL, which contain cholesterol. As an example, let's see how this goes for LDL. Now, on the surface of the cell membrane, there are indented pits that have specific receptors for molecules like LDL. These pits are covered on the intracellular side of the cell membrane by a layer of clathrin proteins, so they're also called coated pits. Now, let's say LDL binds to its receptor in one of these pits. The edges of the pit start coming together. At the same time, the clathrin proteins inside the cell link up to one another like a sturdy shell around the forming vesicle. Once the vesicle pinches off from the cell membrane, the clathrin proteins detach from it and go back to the cell membrane. Inside the cell, the vesicle merges with an organelle called the endosome. Endosomes are similar to lysosomes in that they also fuse with ingested vesicles, but they can also do something else. Endosomes can separate the LDL particle from the LDL receptor it bound to. This is because the endosome has a proton pump that uses ATP to generate a low pH within, which causes the LDL to separate from the receptor. At that point, the vesicle splits into 2 vesicles, one which has all of the LDL that's been brought into the cell, and the other that has all of the LDL receptors. The LDL-filled vesicle goes to the lysosome for digestion, while the one containing the receptors goes back and releases the receptor back on the surface of the cell membrane. This is called receptor recycling because now the LDL receptor can bind to another LDL molecule and the cycle can repeat itself. It's kind of like washing and reusing a dish instead of buying new plates for every meal. Now, exocytosis, on the other hand, starts deep within the cell, in an organelle called the Golgi apparatus. This organelle takes the proteins, lipids, and hormones that are generated in both the rough and smooth endoplasmic reticulum and packages them into a vesicle that can be zip-lined around the cell using the cytoskeleton. The cytoskeleton is made out of proteins like microfilaments, microtubules, and intermediate filaments, which all provide structural stability. The cytoskeleton is also very dynamic, allowing the cell to change shape by selectively extending and contracting the filaments, which is important in some cell functions like muscle contraction, cell division, and even cell movement. The cytoskeleton also helps structures within the cell move from one area to another. Now, there are secretory vesicles which move molecules out of the cell with help of motor proteins like kinesin or dynein, which pick up the vesicle and carry it towards the cell membrane along microtubules using ATP as fuel. The vesicle moves toward the cell surface, fuses with the cell membrane, and ruptures on its external side, spilling its contents into the extracellular space. Alright, as a quick recap. Endocytosis refers to the process in which cells engulf extracellular material, and there are different forms of endocytosis like phagocytosis, pinocytosis, and receptor-mediated endocytosis. Exocytosis refers to the process in which cells expel material into the extracellular space. Both endocytosis and exocytosis require ATP in order to happen.
The osmosis does a great job on this topic again and really can help point that out. All the particulars that we would need to know regarding the endocytosis and the exocytosis here. These will come into play as we keep going through the course. We will refer to this again. Then we went into regulated exocytosis, excuse me. And then when we talk about that, again, diffusion. So facilitated diffusion allows the movement of those hydrophilic, so they love water substances, to go across the membrane down their concentration gradient. Common mechanisms of glucose entry into the cells is through facilitated diffusion. Intracellular glucose is maintained at a low concentration, so as glucose is phosphorylated when it enters the cell and is quickly used for metabolism and energy production. When we talk about the active transport, again, it's important to remember that this means that we need energy to be able to do this. It operates continuously in all the cells, and they use hydrolysis to power transport of sodium and potassium against the concentration gradients. This is the sodium-potassium pump. Very important. It's responsible for differing composition of intracellular and extracellular fluids. So very important there as well. Remember that our primary active transport is going to be our ATP, sodium potassium ATPase. I would recommend that you're comfortable with understanding the active transport of the calcium, potassium, and hydrogen that's talked about in your book. Remember that the sodium-potassium pump is active continuously, but there's also the two additional ATP requiring transporters that are going to be referred throughout the book. And the first is the calcium ATPase, or the calcium pump. Another name for that protein is the sarcoendoplasmic reticulum calcium ATPase, which is also referred to as SERCA. The pump is found on the cell membranes and on the membrane of the endoplasmic reticulum. Okay, so kind of review that part there as well to help us understand those concepts of the other types of transport. The book goes into the secondary active transport, which is co-transport or counter-transport. That's outlined as well in your book. Again, be comfortable with the idea of what they're trying to say, what it means, those kind of things, so that you are just familiar. If someone said, give me an example of a definition of secondary active transport, you can say, that a solute can be transported against its concentration gradient by proteins that link the movement to downhill transport of another solute. Aquaporins allow the polar water molecules to diffuse freely across cell membranes from areas of low osmotic strength to areas of higher osmotic strength. And that's what we can see here in this illustration, that those water molecules are able to transport across the aquaporin proteins. And this is important for the regulation of aquaporin proteins that contribute to the kidney regulation of our body water balance. So when we talk about renal issues, we'll hear again about the aquaporin and how that transport comes into play with our kidneys' ability to regulate our body fluid, how it's going to keep fluid or get rid of fluid. Ion channels are transmembrane proteins that act like a pore. So when the ion channel is open, the ions can freely diffuse downhill, but then they can be closed again. So it's like a gate. Most channels are selective for one ion. So you have a sodium, potassium chloride, or calcium channel. And you've learned about this in the past. I'm sure I'm not sharing anything new with you guys. This is hopefully review from previous days in chemistry. I did want to point out that in your book on table 4.2, they give a nice table here of the transport mechanisms that are overall described in this book so that you can have those as an example as well. So A on this picture here refers to if fluids with this composition are separated by a solid membrane, no ion movement will occur. Okay, so on the left there, nothing's happening, it's solid. But then if you go to the right, if the potassium channels are inserted in the membrane, potassium will move then from intracellular to extracellular down a concentration gradient, and that would generate a membrane potential difference. So the inside would be negative with respect to the outside. So this is a great illustration of the resting membrane potential and how that looks. Then after that, your book jumps into the next topics that we will talk about in part 2 of chapter 4. Feel free to go back and listen to these osmosis videos as many times as you want. I think they're excellent and they do a great job of walking us through this material in an easy way with some nice illustrations. I'll see you in part 2.